So today our speaker is Dr. Renato Borras-Chavez. Renato received a bachelor's degree in marine biology at Andres Bello University in Chile, a master's degree at San Diego State University in California. He worked on kelp forest ecology for his master's degree and then began work on marine mammal science for his PhD work, which was done at the Pontifical Catholic University of Chile, which he completed, I don't know exactly, but a few years ago. His presentation today is was part of his PhD work. So Renato has worked in the Antarctic on fur seals, also done some work, I understand, on leopard seals. And he's worked for 3 years, more recently with the Chilean Antarctic Institute, where he's continuing his work on Antarctic pinnipeds. Currently, he's a research associate at the Center of Applied Ecology and Sustainability in Chile, but he's also preparing to start a postdoctoral project on leopard seals, and I had an opportunity to talk with Renato a little bit before this, and it sounds like he's got some really exciting work ahead, and that leopard seal work will be done at Baylor University. So today's presentation will be on aspects of lactation in the Antarctic fur seal. Antarctic fur seal mothers, like all odorized, give birth on land and shortly thereafter began making foraging trips to sea, returning to land to nurse their pups for a few days, and then continuing this cycle in the case of Antarctic fur seals for about 4 months. Renato will be telling us about differences in foraging behavior between lactating and non-lactating females that have been instrumented with microprocessors to obtain information on diving and foraging behavior in an effort to better understand the challenges facing fur seal mothers. So the paper is based on a paper that's currently in early view. The paper, I believe, has the same title as this talk, Time and Behavioral Adjustments to Lactation, Insights from a Marine Predator. I won't read the list of co-authors because it's substantial, but you can go online and see the paper. If you are not a member and don't have a subscription to Marine Mammal Science, the paper will be available to be read online for the next month. I will put the link to the paper in the chat for you to pick up if you don't have it. So with that, I'm going to turn it over to Renato for his presentation. Renato?
Thanks, Daryl. Thank you, everyone, for being here. Thank you, Daryl, and the editor panel for selecting this project. I was quite surprised with the invitation. That was a great introduction too, so I really don't need to say much other than thanks to my co-authors. This will be presenting hard work of everybody on this list, so I really appreciate all the help. And like also Daryl pointed out, this was part of my PhD, which it has a similar title, A Link in the Fast Lane. We were exploring foraging ecology of Antarctic fur seals, and which is not really a novel thing to explore, but the novelty was that this colony is distributed at the edge of their breeding distribution. And this kind of brought up some very interesting perspectives for the species to explore. So how did I end up? I'm going to start by explaining a little bit how did I ended up working with this species, and this is because I ran into, this was a serendipity event. I ran into the Antarctic Ecosystem Research Division, which is, for those of you that don't know, is manage and implement the U.S. Antarctic Marine Living Research Program. Their study is focused on interaction between the environment, Antarctic krill and fish, predators that depends on krill and fish, and the commercial fisheries that you can see in Antarctica. And it's led by George Waters, which I wanted to also thanks for the opportunity that this division gave me for pursuing my PhD with them. And like I said, it was they provide this science and support decision making by the Commission for the Conservation of Antarctic Marine Living Research, which is the CAMLR. I was on a Chilean vessel when I met Mike Goebel, which it used to be the Pinniped program leader, and he became my advisor on this. We hit along in this boat on those dark times that I was working with Kelforth. No. And I also met Douglas Krause, which is also now currently the program leader of the Pinniped sort of Pinniped departments, and they start telling me about this amazing place that is Cape Giraffe. Is one of the sessions that they are working on long-term monitoring. This is a picture of the refugee where we were staying, and Doug recently was commenting, and I think there is a link around there that this will be replaced by a very fancy, brand new modular alternative as a refugees, which is it has some mixing feelings because it was a beautiful place anyway, but this is probably gonna open some new doors for new researchers as well to be able to explore this place. And if you turn around from this view, you turn around, you will see an even smaller refugee, which is from the Chilean Antarctic Institute. And the reason why I'm showing this is because it's a very interesting process. The Chilean Antarctic Institute start exploring the value of this place in early 80s. And then around 97, 98, which is when the NOAA program started, the US took cover. But during all this time, there's been a really, really nice collaborative work with the same aims of providing quality science for CAMLR. So this is the reason why Chile and the US in this aspect are so connected. Cape Giraffe is located at here in Livingston Island. You can, let me see if, yeah, here you can see that square there, and that's Cape Giraffe. And it's a very unique place because if you helicopter to Cape Giraffe, you land it and you'll see like pure white. This is a picture from probably November. But then when you start walking around, you will see this amazing biodiversity that is being held in this place. You can see most of the species of pinnipeds that are found in Antarctica, other than the Russell. I've seen them all, but you have some reproductive breeding areas of 2 species of penguins, gulls, and many other birds that are nesting there. If you're not a big fan of megafauna, you also can find the vascular plants, the only 3 species of vascular plants that you will find in Antarctica, and some other paleontologic, paleobotanic elements as well. So anyway, it's just an amazing hotspot, Antarctic hotspot, and I wanted to focus on the one of the most abundant, although declining, but one of the most abundant animals there that is also breeding, which is an aggregation of Antarctic fur seals, which are central place foragers. For those of you who don't remember that, it's central place foragers are defined like organisms traveling from a home base to a distant foraging location. And Antarctic fur seals during lactation, just like Daryl pointed out, females make multiple trips to sea to supply upon return to the breeding beach the energy needed by their offspring. So when females are foraging, pups fast usually between 1 to 6 days. In this species, depends on the location, but with some other fur seal species reaching way beyond that. I've been working in Juan Fernandez fur seals, and they can reach up to 30 days that the pups are there waiting for the mothers to come back and get the milk that they need. This is not an elephant. This is the shape of the Western Antarctic Peninsula. And if we talk about the distribution of the species, most of the individuals are found in South Georgia, with, I think last time I read, it was over 6 or 7 million individuals over there. But Cape Giraffe is sustained the largest aggregation of the Western Antarctic Peninsula as well. And when we start exploring what we know about foraging of this species, the most important prey is krill, and followed by a couple of myctophid species. And you can also see, depending on the location, what is the role of squids as well in this diet. But it definitely krill is one of the most, is the most important of prey that they capture. They can reach 150 meters depth in regular diving, and on average they dive 13 to 20 meters. A lot of the diving occurs during the night. The trip duration, it's about, on average, depending on the location, but it's 4.5 for 3 to 4 days. The mean dive duration is 1 to 2 minutes, and these are more some difference that you will see with the species compared to other pinnipeds. And the percentage of time diving of the entire trip is usually between 20 and 35% of the time. So foraging behavior of this species, just like many like this one, it can be separated in 4 aspects: or the horizontal displacement, where are they going, the vertical component, which is diving, and the habitat use and the prey that they capture if they are successful on this. And these aspects are shaped by intrinsic and extrinsic factors, while foraging adjustments in behavior to balance energy acquisition and pod provisioning can occur, for example, when traveling, or when provisioning, or even diving. Many extrinsic factors are shaping the foraging behavior of Antarctic fur seals. This is one of the most well-known species, fur seal species in the world. The use of different foraging areas due to, for example, changes in prey availability, environmental variability affecting prey, or when they face scenarios of limited resource against those that are not. Behaviorally, Antarctic fur seals are responsive to these extrinsic factors. For instance, under constructing scenarios of prey availability, lactating females will modify their trip duration or the time spent ashore. So in years when prey availability is limited, Antarctic fur seals will adjust their time budget. Basically, trip duration would be limited by the availability and abundance of the prey, but the time ashore, the haul out time, will be limited by rate of energy that is transferred to the offspring. And when it comes to intrinsic factors, we can name a few. Fur seals female age is different because of the experience of these animals on knowing the area where they are foraging, the status, the age, the mass, even the colony size have been shown to play also a role, and the mass of the pup is not the same to take care of a tiny pup compared to a 15-kilogram pup, for example. So when I was talking about the project with my Chilean advisors, because I got my PhD in Chile, which they come from a non-marine mammal background, from Dr. Francisco Bocinovich and Jose Miguel Fariña. And the first fast question was like, oh, okay, this sounds great, but what about the control group? And I really didn't think about it at that time, and it makes a lot of sense for them, because they say, well, all the animals you're mentioning are lactating. So if the lactation, we know lactation is the most expensive reproductive event of a mammal, you will need a control for that. And actually everybody knows this. Even I have a friend yesterday, she saw a news article related to this paper, and she wrote me back like, oh, I used to eat everything when I was lactating. So this is not just something related to fur seals, but this happened in every single mammal. So I start digging into it. In the case of Antarctic fur seals, you will have that John Arnold in 97 suggested that lactation can account for 31% of the energy expenditure. And I really didn't think about it before. So I look it up, and in 1997, at least no diving records of non-lactating females were there. And 20 years later, when Paul Pongianis published his book, there was still I realized that this was not just happening with Antarctic fur seals, but also like otoliths. There was very limited information on diving behavior and lactating individuals. These have changed this last few years. There is still a little bit more, but it really kind of shaped the question, the research question. And do we really need this control? Do we need that information? Well, let's check an example. In years when prey availability is limited, Antarctic fur seals will adjust their time budget. So females will reduce the time spent ashore and increase trip durations to maximize energy delivery. In contrast, in years when prey is abundant, females would perform shorter trips to sea. Okay, so we argue that the short trips observed in lactating females under abundant prey scenarios could also be associated with the constraint of lactation or a tendency, and not solely shaped by the prey availability. So the use of only lactating females in previous studies does not allow for a clear estimation of the additional work needed by breeding females. In other words, we understand the energetic cost of lactation, but we could not say what behavioral adjustments are needed to cope with this expensive event. And this is again Cape Giraffe, and it gets really even more excited if you think that animals at Cape Giraffe are operating at their southernmost limit of the species' reproductive distribution. So in years of low prey availability, Antarctic fur seal females increase foraging trip duration, but because females are operating at their maximum capacity, under no circumstances would they modify their diving effort. That's what we know so far. So again, the question is it possible that this additional effort, that you can see it in the diving activities, is being masked by the fact that we are only measuring effort over lactating females? Increasing effort have been observed in other pinniped species under similar demanding reproductive events, such as pregnancy. Of course, an energetic difference between gestation and lactation notwithstanding, but we see that animals in general are responsive on behavior with that. We also learned this from the few comparative studies, mostly in terrestrial mammals, that exist, that the only way of finding the actual adjustments associated to lactation is by performing simultaneous measurements between lactating and non-lactating animals. So the research question was, are there differences in the foraging behavior between lactating and non-lactating females? We tested the hypothesis that non-lactating individuals perform longer trips to sea and have longer times hauling out than lactating females due to the absence of restrictions associated to lactation. And in addition, given the same foraging environment, lactating animals would show also additional effort by either increasing their diving activities as a result of the additional energetic cost associated. So those two were the hypotheses that we were trying to test. And we went to Cape Giraffe as part of this monitoring program. They have everything very nicely standardized. So, and this specific experiment was conducted on the reproductive seasons 2015-16 and 16-17, which I'm now going to start calling only 2015 and 2016, which use non-age females of relatively similar size. And then we anyway calculate the body condition index, because there are differences in mass because mothers are lactating as opposed to the other ones. But we try to maintain a relatively similar length, at least standard length. This part of the thesis included only those trips that we were capable of comparing simultaneously, because instruments on non-lactating animals last less, a shorter time than what it was for non-lactating animals. So we separate these 94 trips of 14 lactating, and we compare them with these 27 trips of 5 non-lactating. We managed to tag 7, but we could not, we lost 2 of the instruments. So that's when we start noticing, oh, okay, maybe this is why nobody was doing it. We also tagged the pups, and this is also part of the protocol. And we took milk samples for diet and analysis. And we installed these instruments, which are TDRs, time depth recorders, that they can measure depth, temperature, and light. For those of you that haven't worked with TDRs, it's very straightforward. You can see here that you have on the y-axis depth and then date, and each of these lines is a dive. So this big group of lines would account for the entire trip to sea, whereas these where you cannot see those lines, it would be the time that they spend howling out or back in the breeding area. So just by having these, once we filtered the data, and this was done by an Ignos toolbox developed by Ian Tremblay, we can sort of correct the surface, make a zero offset correction at the surface and identify dives based on a user-defined minimum, which was 4 meters and 6 seconds. And just right away, we can easily see how long the trip durations and the hallow time is for these animals. We can also calculate the dive rates within each of the trips, and we validate the trip duration and hallow time by also using VHF data transmitters and daily visual observation. So there's people that goes every single day out and run and walk the beach and see if these animals are there, just to validate the information that we get from the instruments. Now, if I take one of these lines and I sort of zoom in, I will see that we can also get some characteristic of the dive itself. We can get the dive duration, we can get the bottom time, which would be the bottom phase where most, it's been described that usually feeding happen, and maximum depth of these animals, and also the vertical component, which is the descendant time, descendant rate, ascendant time, ascendant rate. So all these different diving variables were also going to be compared between these two groups that we had. Metrics of diving behavior calculated at the scale of the complete foraging cycle. For example, mean dive frequency, the dives per hour that they do. May mask also differences observed within individual dive bouts. The dive bouts are a group of dives interspaced with surface time interval. So we also wanted to explore how the bouts were being configured, and we characterized many other variables that were related to these bouts, but I'm not going to talk about this because we didn't find any difference. I just want to point it out that you can see some of this information on the paper in the supplementary material. Previous studies have also described that there is a variation in the vertical daily migration patterns of the prey, which will result also in animals diving differently. So to explore this, we assign foraging dives to whether they happen during the day or night, based on the sun angle at the time of foraging. And I'm also going to show you real quick the results because we didn't find any difference. Most of the animals in red, you have the lactating, in blue, the non-lactating, and 53 to 54% of the time they are diving in the night, which is still a lot more than during the day, considering that night is in Antarctica, it's a lot shorter at this time of the year. We evaluate if every single foraging variable was explained by group meaning lactating or non-lactating, and so we built linear mix effect models and GLMS, and we use the season also as a fixed factor, whether the animals were a group would be the animals lactating or non-lactating. Also, we incorporated the body condition index and also the interactions between these variables as fixed factors. Each identity of each female was also used as a random factor. And then when we were talking about diving variables, we also added as a fixed factor whether the dive occurred during the day or night. So then with backward stepwise modeling selection, we use the ACAC information criteria to see on each of these foraging variables which ones were explaining the variance of that was being found, whether some variables were different or not between groups. Because we didn't have any control of prey availability, which could also shape differences between breeding seasons, we also characterize at least the prey being consumed by analyzing 100 scats per season, which is something that also the program does every year, and in account for like 10 scats per week during the entire breeding season. So here we can estimate occurrence of these animals. We don't quantify how much they eat, but we can tell. For example, in pink you have krill, so we know that krill was almost all the time present on the scats of these animals. And fish, you can see that they start showing up a little more right in the middle of the breeding season, like by the end of January. So we can identify some sort of a exclusively krill phase as opposed to a fish krill mix phase during the breeding season. And 95% of the diet was krill. The occurrence of diet was krill and two species of nyctophids. We also compare the size of the krill between those 2 years, and they were relatively similar, more variables 2015, but relatively similar size of krill. So this kind of told us that the years, this in the way of characteristics of this prey were similar. And these are some of the results. In this graph you could see hallow time, and these are the 5 non-lactating animals with the trips that we included of each of them, and these are the remaining lactating females. And the first thing we can easily see is that non-lactating animals are spending almost 2.4 times more time on shore than lactating animals. So we already have double the time that they're spending back when they go back to the breeding areas. That was right away our first conclusion. Lactating females spend less time ashore than non-lactating animals. If we take a look at the trip duration, here again, lactating is in red, non-lactating is in blue. Super simple. We can also see that trip duration was shorter for lactating animals, but this, in this case, as opposed to what we saw in hallow time, this was driven mostly by the differences found in one of the years than the other. And in 2016, there was like a big difference between 5.7 days compared to 2.9 for lactatings, whereas in 2015 we found only very similar, like 3.5 days, regardless of whether they were lactating or not. If we plot the hallow time and y-axis and the trip duration, we can also see that regardless of the duration of the trip, you would always have more time on land of howling out of animals that are non-lactating compared to lactating animals. So the first hypothesis was pretty straightforward. We found that non-lactating animals are indeed spending either longer time some years, or at least it's very variable. We also checked, we did check the variance of these trips, and the variance was a lot bigger in non-lactating animals than what it was in lactating individuals. So they can do either, for example, here's a good example. We have the instrument of one specific non-lactating animal. She did a total of 4 trips, a very long trip, and then you have another animal that is capable of doing very short trips all the time. As contrary to this, most of the animals that we got on lactating were very, very structured in the way of doing all the trips, very short trips and consistent through time as well, as opposed to non-lactating. So some conclusions of this trip duration differences were driven by non-lactating females, and specifically longer trips in 2016. And we also found some variance being explained by the body condition index, which it's more likely to be related to the mass differences. I'll talk about it a little later about that. In 2015, both groups show similar trip duration, which is around 3.5 days. And the mean hallow duration of non-lactating females double the time spent on land by lactating females. And also very interesting, regardless of trip duration, lactating females spend less time ashore compared to non-lactating females during the entire period that they were monitoring. Now, when it comes to the diving, which we thought we were hoping to see some sort of the effort, lactating animals show shorter dives. And it happened mostly reducing this dive duration was mostly reduced, explained by reducing the bottom time of a dive. And the bottom time of a dive is not always a good predictor of foraging success. It was thought about that, but its recent publications have shown otherwise. It really depends, especially for those that are diving more close to the surface. So reducing the bottom time of a dive might not substantially affect energy acquisition. And in addition to that, we also find that descendant rate was was playing a couple of additional seconds of difference. They were also reducing the descendant time. And the statistical difference between groups, I mean, we found also statistical difference that were explained by, again, the body condition index. But we got to be careful with the body condition index, because this was calculated based on when we install the instrument, which is at the beginning of the season. So we only have a very tiny snapshot of the weight of these animals, the mass of these animals, that is representative of when they arrive to Cape Giraffe. So just be cautious on making big conclusions about it, because we didn't have the possibility of capturing many times the same animals to be measuring the mass of these animals on an extended period. What we believe it could happen is like to be able to reach the same depth for lactating females against a non-lactating female. Lactating females could be performing steeper dives than non-lactating females. And with this, they will also, the benefit of this is that they will reduce the metabolic cost of a dive without changing the descendant speed, which we didn't find any change on the descendant speed. This behavioral strategy has been observed in Antarctic fursuits performing steeper dives to compensate for slow speed dives. It has been also observed in penguins, in many species of penguin, that they reduce the descendant time of dives by doing steeper dive angles, which reduce the cost of transportation. We propose that when diving, lactating females minimize the cost by reducing diving time rather than maximize energy return per dive. And by performing shallower dive, this strategy specifically shouldn't impact foraging success substantially. But of course, this is something to be tested anyway. To prove this hypothesis, we need to compare metabolic measurements coupled with diving records between non-lactating and lactating females, especially considering our sample size of non-lactating animals. And the difference in diving behavior could also be due to buoyancy or drag effect. We know that in faucets, difference in buoyancy influence diving behavior, specifically modifying descendant and ascendant rate, the vertical phase, to be able to maintain vertical speed. But in otoliths, drag seems to be having a higher impact in dive performance than buoyancy. We try to control size as well as age, like I mentioned at the beginning, but there has been there's evidence that even minor differences in mass, like 250 grams, for example, can reduce the time that females spend diving significantly. And actually, Gita in 2009, McDonald and the collaborators found a reduction in dive effort in larger females compared to smaller ones in the species, which is consistent with our findings, where sort of heavier lactating females show shorter dives compared to lighter non-lactating animals. Again, we need to be cautious with this, because we will need to measure the mass of these animals and how it changed through time to be able to actually say this. But it's a good exercise to see that to see other potential alternatives why we're seeing this variability in diving behavior. Another proxy that we used for diving behavior was to estimate the percentage of time invested diving, and this was very interesting. In the top here, we can see 2015 and 2016, and the mean percentage of diving time per trip for lactating female was in 2015 10% higher than non-lactating females, but it was very similar in 2016. So this kind of start telling us like, okay, so there was something going on in there was a difference on the years anyway, which is also sort of included in the models when we are acknowledging that season could also have an impact on the way these animals or the differences that we could find in these animals. So non-lactating females might consume, for example, more fish than lactating females, and reducing the time invest capturing krill by increased consumption of relatively more energy-rich prey. And in contrast, the mean percentage of diving time per trip did not change at all. Like if we see lactating females here and here, regardless of the year, they are very similar percentages. And I'm going to expand about that a little bit later on. But our second hypothesis is that they are increasing the time invested diving. Yes, we can see it proportionally, but at a very at a very specific level on each of the dives, we're not seeing that. We are seeing the complete opposite. They are reducing the time per dive by extending the total time. So lactating females are constrained to spend a similar proportional time diving regardless of the prey available. Trips considered in our study, and this is why I want to stop here a little bit, because trips considered in our study differ temporally on each season. So the prey available at the moment of the trip could have also be different. They are simultaneously measured, but the 2015 with the 2016 are not necessarily in the exact weeks that this happened. And I can pull back the scat graph because I can show you here with this black bar here shows the time frame of the trips compared simultaneously per year. So in other words, the marked difference in the percentage of diving time observed in 2015 may be related to differences in the prey target, because at this location, we know there's a well-documented increase in the incidence of fish in the diet after around the end of January, whereas the foraging trips evaluated in 2016 represent more this first half of the season, which is mostly we observe in the diet that it's almost exclusively krill. So maybe lactating and non-lactating animals are targeting the same prey here as opposed to what they're doing in 2015. Now, alternatively, what we did not consider is that there might be difference in the abundance of prey between years. And it's really hard to get this information because I remember people from NOAA was telling me that back in the days, they were putting instruments, but also there was a vessel checking these areas where the seals were going, and you have a transect which was kind of telling you how abundant the krill was in these areas. Well, we didn't have that for these 3 years, but we come out with an idea of and we took a krill data from the long-term Palmer Station Antarctic program, LTER program of NSF, and they perform annual zooplankton surveys during January, including krill within a grid area that started approximately 250 kilometers south of Cape Giraffe and extends southward. So some of the sample points are representative of the downstream pathway flow existing along the Western Antarctic Peninsula shelf break that also kind of cover the Cape Giraffe area. So difference in krill density were statistically significant between these years. So yes, we have similar characteristic of the krill, but that doesn't mean that it's abundant. And so this is density numbers of individuals in 1,000 meters square of water, and they were like there was a higher mean density in 2015 than 2016. Again, this suggests that giving a reduction in krill abundance in 2016 relatively to 2015, the time invested diving for both years by lactating females, which it was almost around 25% in both of the years, might represent the maximum percentage of dive time that lactating females can invest at this location, whether the prey is limited or like the scenario of 2016 or not, which is 2015. In other words, lactating females may be incapable of increasing the percentage of diving time per trip, even if prey is less available, limiting the behavioral response that lactating females can have at this latitude, at the edge of their distribution, when facing specifically complex scenarios of prey abundance. We certainly need to measure this off of Cape Giraffe if we want to be sure that this is the case in these areas. But here again, we are trying to provide of multiple alternative hypotheses that could be explaining the differences that are being found anyway between these two groups. So as a conclusion, lactating females behavior is characterized by shorter foraging trips to sea, shorter hallow time, and the data described in previous studies only when prey availability is not limited. Also, the combination of shorter foraging trips and shorter time spent ashore is consistent with a strategy that could maximize energy delivered to their offspring. I spend very little time, I go back, I give milk, I come back out. That's basically as simple as you can put it. At the same time, like at a fine scale, lactating females might minimize the cost of diet by reducing bottom time and descendant time, but increasing the percentage of time spent diving per trip compared to non-lactating females. This adjustment in diving behavior can also be the consequence of body compositions, like I mentioned before, but we need more data for that. A diving strategy that combines a reduction of dive duration per dive and an increase in the percentage of time spent diving per trip would increase the energy intake per trip. And this has been observed in other mammals under energetically expensive reproductive events. In contrast, like we were saying, non-lactating females, they can perform long and short trips. There is not really an evident pattern here. They do definitely longer haul out times than lactating females. And it's all this, and also longer dives and less time invested diving. It's really the result of a behavior that is partially characterized by the absence of breeding constraints. And those seals that do not have these constraints of pup rearing exhibit greater flexibility in behavior. The main challenge for comparative studies using non-lactating females, it's probably the unpredictable behavior and the fact that they are not obligated to return and to their offspring. So I couldn't find a picture, but I still remember capturing females, non-lactating females on hills, very far from the breeding areas. And it was a lot more, I don't know if they were doing more effort or we were doing more effort by trying to find these animals. And that also kind of explained why this, I never thought that this question was never arise. It's just very risky to put instruments on these animals because it's very likely they can easily just never come back. And that was a risk that I appreciate Noah that took with these questions. So we can solve these questions somehow. And this is why these studies are sort of have a narrow temporal, are compressed to a narrow temporal scale or a sample size that are small sample size. A larger sample size of non-lactating females could provide knowledge about the intraspecific variation, for example, of non-lactating females' foraging behavior and diving behavior as well, over longer temporal scales or, for example, under different environmental conditions. I think a really cool question to explore is that future studies should compare non-lactating versus lactating females, but coupled with simultaneous field metabolic rate measurements. And that would provide definitely additional power to support our findings and a better understanding of how marine mammals respond to rapidly changing environment. And when I say rapidly changing, when I say rapidly changing environment, I'm not talking about the transition of the breeding season. This is an example we can observe here. Like when we get there, this is one of the Wiley took these pictures. That it's very easy to see how rapid change occurred during the entire breeding season. This is when we got there, and then when we leave, it's you can see that the snow is almost all gone, and it's literally everything. When I arrived there, I arrived like this, and then I left the island like this. So everything changed. But we are more important for us is the long-term changes that are associated with the current global change scenario. So, and this is just quickly a promotion of that. This second paper of the thesis is trying to link diet behavior with quantitative prey-specific fatty acid estimations by using machine learning tools, so we can then later on know exactly what these animals are eating only from the archive TDR data. With that, what we found here you have the Livingston Island, again, Cape Giraffe, and every single one of these sort of areas, colored areas, is different trip, and the orange, it's showing you where the animals are foraging. So the yellow arrows, this is the continental shelf break, and then you have the through, which is a remnant of a subduction zone here at Cape Giraffe. So we already know that we found 4 strategies that we can also tell you approximately what these guys are eating. They have 2 strategies that they combine or mix krill or fish. One of them is more oriented to eat krill, and the other one is oriented to eat fish, but they happen around the continental shelf, both of them all the time. But we also have some other strategies, such as these ones, that they extend these their longer trips to even far from this continental shelf break, and they explore, and these are it's more than 70% of what they are targeting here is fish. And we have other areas that they or go also very, very far from what they usually do, which is the ones in the middle, but they are targeting krill. We know that from the way they behave. So this is very interesting for us. We are checking it in 3 years, but then it's going to open a possibility of retrospectively check the information that has been already collected from TDRs. Some of the primary conclusions of this aspect is that we managed to identify and predict prey-specific dives from diving behavior using fatty acid quantification. We characterize foraging strategies at this location, including what prey species are targeted. And like I said, this is gonna sort of open the door and explore the already collected TDR data of the last decades. Yeah, I actually want to take this moment to sort of promote. It was this year paper that you can find out a lot more about the animals from Cape Giraffe. Douglas Krause and collaborators, including a lot of the few folks that I mentioned at the beginning. They came up with this very, very nice paper, in which you can see a lot of other aspects of foraging behavior of these animals, and a lot of data that this program is collecting in Cape Giraffe. And if you are more interested in knowing what is to be for almost 5 months or 4.5 months in Antarctica, I will strongly recommend you to read the upcoming book of my good friend Naira de Gracia. She was a technician at Cape Giraffe for a couple of years, and she's just a gifted person when it comes to writing, and her book about Cape Giraffe, I think, and her experience with penguins, but there are some seals in between, I know for sure. Which I think here's she probably looking for inspiration of writing this book. It's gonna be on sale, I think, in April 2023. I want to thank all the co-authors again, not just for this first paper, but also the one that is coming up, and I think another one that should be coming up soon. But also all the people on the field. People don't realize how much people you need people that doesn't work with these animals don't realize how much people you need for these things to happen, and the coordination and the logistics supports are that was for me it was a turning point in my career. After this, I could not just think in any other than marine mammals, and specifically pinnipeds. So I want to thanks my advisor Mike Gobel and everybody on the team while we were doing there for for changing the path of my life. And I want to thank the funding institutions again. I really appreciate. I'm very jealous of those that are working at NOAA every year at Cape Giraffe. And I also want to thank the Chilean Antarctic Institute, because they fund part of this project, as well as the ANID, which who funded my PhD while in those few years that I did it, including the University of where I am. So thank you very much to the society, to the student members, the small group that organized this. Thank you, the editor, for trusting me on this. And by that, I would love to take any question.
Thank you, Renato, for that interesting presentation. We, at the moment, have one question, It's one that doesn't have so much scientific content as interest in your work. It's from Kevin Lester. It says, wow, I love that depth analysis graph. Do you have a GIS specialist? I work on 3D distribution modeling in killer whales, but I'm a pinniped guy, so I'd love to help you out.
Yeah, no, I definitely need to to improve my skills in that. If I want to start doing better figures in that, I don't. To be honest, we don't have anybody yet. We have been going back and forth, doing better work with that data, and yeah, we can definitely talk. I completely forgot to provide my email here, but please let me just. My email is on the paper as well, so let's talk about it. And between pinniped people, we need to support each other, and it's a good asset what you're telling me that you have. Thanks for your interest.
Okay, and we have one comment from Mateus Castillo Aguilar. Sorry about my pronunciation. Says nice presentation. Congrats. So we
Thank you, Matthias.
Don't have any challenging questions here, but I'll come up with one question that I wondered about listening to the presentation. So your sample size for non-lactating females was pretty small.
Yeah.
You have a lot more variability in some of these variables. To what extent might the differences you find between lactating and non-lactating be influenced by that greater variability in behavior and small sample size combined? Do you think if you had a larger sample size, you'd still find these differences?
I'm more confident on the differences that we are finding related to the attendancy cycle, specifically trip duration and haul-out time. That is exactly why we decided to explore most of the alternatives that exist when it comes to diving behavior. The complexity of the lack of control that we have of the environment, it makes these type of studies very hard. And answering your question, I think diving behavior might just be an artifact of all these prey availability scenarios that we put together, but also the strategy that we came up with is also did make sense, and it's been used by the same species in other areas. So it certainly will either prove us right or wrong when it comes to dive behavior, but I'm pretty sure we have seen in other actually the haul-out time was very consistent in every single trip that we explore in non-lactating animals. So I'm at least I'm sure about that one. And trip duration, my most important result, I think it was not much of the difference, but the fact that there is some limitations on how extensive those trips can be. And and when it comes to the diving behavior, also the percentage of the time that the lactating animals are investing, regardless of the scenario that they're facing, that is one of the really cool things. Because even if there's only a very small sample size of non-lactating animals, it is already proved that they are being more heterogeneous in the way they are diving. We can see that they are spending a little bit of time one year, and then the other year a much longer time diving. But when I compare the lactating animals, this was 14 animals that we show here, but I have the rest of the animals that we use in these 3 years. They are showing very similar patterns when it comes to the percentage of time that they spend diving. So we at the beginning, when we were writing the paper, and we were thinking on kind of writing it talking about the non-lactating animals, but then we realized that because of this small sample size, we were more confident on the result that we're seeing in lactating animals. But the contrasting aspect that was given us, that was being given by this small group of animals, was still very insightful when we were seeing these consistent patterns that we were obtaining or checking on the trip durations of the lactating animals.
Thank you. We have a question from Tiffany Geniard Dudot. She says, hello, thanks a lot for the great presentation. Did you look into the differences in environmental parameters between the two study years that could explain the observed yearly differences?
Yes, not experimentally, but you will see, and I didn't brought it up. Nice reading you. I read your paper, by the way. I read your papers. We had seen 2000, if I'm not mistaken, 2016, the year where we also found very a lot less dense krill on the waters, was consistent with the year. Well, I guess with the year where El Nino influence was ended up in the record temperatures of water temperatures in Antarctica in 2015. And so there is a couple of papers of Atkinson, and he did help me a lot when I was writing my thesis to understand why that year was so different than the other two. And it turns out that indeed there was a lot less krill, and it was oriented also to our southern areas. And this was all explained by the temperature, not the temperature that you have during the austral summer, but the temperature, the previous temperature, which it will have an effect on a spawning of krill spawning, and later, of course, it will affect the availability of the prey in that season. So it was very, we check it out by for the sake of discussion, but we did not incorporate it in the analysis.
Okay, we have no further questions there. Let me just ask you then one more question of my own. So given the lactating females showed no difference in percentage of time, I don't recall, diving, I think is what it was, percentage of time diving, between the 2 years, despite the considerable difference in prey density. Do you think that potentially your study area is an area that's sort of marginal for these fur seals? Do you know how that percentage diving compares to other areas where the Antarctic fur seals are breeding?
Marginal in what sense? What do you?
Meaning that they're essentially working as hard as they can to get their resources for sustaining lactation.
Yeah. Yeah, I strongly believe that. And this it started with I think Dan Costa did a great work in 1989 where he was showing energetically that they were operating at their maximum capacity, and then he explored this further in another paper of 2007, in which he compares many species of fur seals and how they are reacting, and it turns out that, and how they respond to environmental variability. And it turns out that the Cape giraffe colony was one of the examples of just operating at their maximum capacity. And my experience with the Chilean Antarctic Institute also allowed me to go even farther south. And so I have been taking advantage of being on a vessel with the Chilean Antarctic Institute, exploring different areas all the way down to Margaret Bay. And every single animal that we have observed there are either juveniles or males, but we have never seen pups or breeding areas around that place, even though there is enough literature that Margaret Bay, for example, has their own I mean, you can find a lot of krill. It's another hotspot of this area. Of course, site fidelity of these animals might be playing an important role of why these animals are not really moving from there, or it's just simply even if you have available prey southern, that doesn't mean that they're gonna you will find more animals there. So I strongly believe that these animals are indeed operating at their maximum. And even if you find like very small areas with a couple of pups, it will be I heard somebody telling me that there is a paper coming up about a very, very tiny colony that might be starting to be populated by a breeding colony a little southern than Cape Giraffe, but it's still I think it's still the most important southernmost colony anyway, and it's restricted by not necessarily I think it's restricted by the energy constraints. When you add up lactation and environmental variability existing in this place, a paper is coming up by a colleague from Spain. It's going to be really interesting because he is exploring males and females, winter and summer patterns of distribution in Antarctica under different climate change scenarios, including environmental variability and environmental multiple environmental variables. So it's going to be really cool because then we can see at least where we are projecting that these animals could go, even though maybe they will be restricted by, for example, site fidelity and other aspects of their own biology. So that's hopefully will be coming up next year, I think.
Okay, great. Thank you very much. Well, I don't see any further questions. We're just after 2 o'clock, so I think we'll call an end to the seminar presentation. Really appreciate interesting talk, work that's very much of interest to my past research on pinnipeds.
Yeah.
So, thank you, and we'll go ahead and sign off at this point. And I look forward
thank you
to
very much, Daryl. And
yeah, I look forward to your leopard seal work, and keep Marine Mammal Science in mind when you get around to publishing it. With that,
of course, always. It was a pleasure publishing with you, and thanks for your insights on the review. The review process, the whole thing was just I learned just a lot. So I also recommend it for that. It was a really nice process. So thanks, and thanks for the invitation. Bye bye.
You're welcome. All right. Bye bye.